Thank you everybody for joining us for today's Substance Use Program of Research CFAR seminar. We're very pleased to have our guest presenter today, who is Dr. Pamina Gorbach. Dr. Gorbach is a professor in the Department of Epidemiology in the Fielding School of Public Health and the Division of Infectious Diseases in the Geffen School of Medicine at the University of California in Los Angeles. Dr. focuses on biobehavioral epidemiology of infectious disease, especially how HIV transmission, acquisition, and progression is affected by substance abuse. Dr. Gorbach is MPI of a research platform entitled Minority Men Who Have Sex With Men Cohort at UCLA, Linking Infections, Noting Effects, which is also known as the mSTUDY. This study has established a Los Angeles cohort and biorepository to support research on the intersection of HIV and minority men. She also heads the Biobehavioral Assessment Research Center at the Fielding School of Public Health, and she is the co-director of the UCLA Center for AIDS Research's Translational Research for Substance Use program, as well as the UCLA T32 for the postdoctoral trainees and ID fellows for biobehavioral research training for ending the HIV epidemic. We are very pleased to have Dr. Gorbach join us today, and I'm going to pass the mic to her. So everybody, please give her a great warm welcome to UCSF virtually.
Hi. So let me see if this will start it as a slideshow. There we go. I'm going to talk to you today about the mSTUDY that was in the intro. Let's see, it's doing it. I don't like it when it does the side view. So hang on a second. Let me adjust the version of how you do display settings. Here we go. I think that's it. How do I make that? Do you guys see the next slide stuff on the right, or am only I seeing that?
Yeah, we do. I think in the display settings, you could do swap, Pamina.
Okay, let me see if it swaps and it takes it out. Oh, it's okay, let me try a little bit better to do it this way. I'll try it one more time and then I will. There we go. Okay, so today I have to tell you that I took a huge pivot yesterday, and I was going to talk to you about one of my recent analyses on pain and the experience of pain among young men that I've been working on. That's a manuscript we're about to submit. And then I realized that this is my first opportunity to actually review the whole decade of work that we've done on the mSTUDY. And so literally yesterday morning, I suddenly decided to do this grand overview for you all and pull it all together and do some thoughts for myself about sort of what have we done in the past year. Whoops. And where are we? And so I have done that. And so bear with me a little bit if things mess up, but I'm going to try to take you on a grand tour of the mSTUDY that is reaching its 10th year this summer. So I'm going to talk to you about the background of it. I'm going to give you a little bit of the infrastructure just so you understand the platform of the mSTUDY itself. I'm going to then give you some focused findings. I'm going to show you the summary data and then talk to you about sort of where we're pivoting and the evolution of where we are now. And I welcome questions. And so since I put this together a little bit late, I'm happy to have you interrupt me as we go and do this a little bit more informally. So please jump on in with the chat or with raising your hand saying something. So what is the mSTUDY? So the mSTUDY is a NIDA-funded research platform. That means it's a U01 that since August 2014 has this cohort of over 500 HIV positive and negative young men who are identified as men who have sex with men. And we're mostly men of color. And we've been enrolling since 2014, although funded since 2013. So we enrolled the cohort to intentionally have half who were people living with HIV and half who were actively using substances at the time of enrollment. The mSTUDY enables important tests of biological influences on substances, on immune function to investigate the links between initially non-injection substance use and HIV. And that was intentional because at the time that we started, there were many cohorts. Some of you are probably familiar with the ALIVE cohort, the VITAS cohort, JHCC, other cohorts that are in actually the consortium we belong to of these NIDA U01s, and they focus on injection substance use. So we specifically wanted to focus on non-injection. We've actually removed that as things have evolved over 10 years because we have people who inject stimulants. That's our focus. But initially we were formed as a non-injection substance use cohort. Cohort participants have visits every 6 months where they donate specimens and they fill out questionnaires, and I'll give you more detail on that. And then we have this extensive biorepository, and I will give you also more details on that. So the objectives of what we're doing in the mSTUDY are to assess the impact of substance use, particularly stimulants and cannabis, in a diverse group of mostly men of color who have sex with men on HIV outcomes. The outcomes include HIV prevention, so that we do have quite a bit of work on PrEP, HIV disease progression, comorbid STI infection, and mental and general health disorders, as well as socio-behavioral health disparities. We have extensive biological sampling, which allows for basic science research on the effects of substance use on the mechanisms of HIV. And we do get a lot of involvement and feedback from our community partner, particularly the LGBT Center in Los Angeles, that allows the mSTUDY to maintain its high level of, my screen is blocked by this bar, of relevance to our population as the epidemic evolves over time. So let's take a look at, where we do this. So the mSTUDY has 2 sites. We are a community cohort, and we have all of our people living with HIV followed at the LGBT Center. This has been my partner for over 20 years. I have done many studies with them. But in this study, they are such a key role. I can't say enough about the wonderful collaboration we've had. And they really run most of our recruitment across the entire cohort. If you don't know about the LGBT Center in Los Angeles, it's part of a large organization that actually is broader than just health services. It includes also social and housing and other supportive services, and it has a HIV clinic called the Jeffrey Goodman Clinic. We also run this at the Vine Street Clinic, which is a UCLA research clinic. It's our only outpatient research clinic that was actually established by Dr. Shoptaw and I in 2006 for another NIDA cohort way back in the day, but it's continued to be a clinical research site. It does HPTN and HVTN studies, and a lot of clinical trial research on substance use. And it follows mostly our people without HIV. Both sites have full labs, nurse services, phlebotomists, and 3 dedicated research staff. So where is the cohort now? We have enrolled over 656 people. We have about 9.5 years of follow-up data on them. We are coming on our 10th year this summer. We have almost 5,000 study visits, 4,975 visits. We had 345 people living with HIV and 311 without HIV. They're not all still in active follow-up. Partly that is because we have had 18 seroconverters, which is an incidence over the 10 years of 6%, which is pretty high. That's over 10 years. And we've had 23 known deaths. And actually, we are right now digging into the death records because some of our people that are lost to follow-up, which is not a high number, but we do have some, we are trying to see, and we anticipate, because we just found, I think, 7 more in the people without HIV that have actually passed away, that have died. And I can tell you that almost all these deaths are due to overdose. We have some that are due to complications of HIV. So I'm not going to go over this in detail, but this is just to let you know, as I said, at every 6-month visit, we have a extensive questionnaire. And the questionnaire is done through ACASI, and these are the blocks of the different topics that we study. We have moved these, some of these out of rotation, and some of these, as you can see with the double stars, are not done always at every visit. Some are done only once, and some are done in rotation. But we have lots and very detailed behavioral data on a whole broad range of topics, and these evolve. And that's why we moved into these blocks, because some things have become less critical over time, and some things have become newly salient that we have needed to expand the behavioral questionnaire. And this is always a challenge with the cohort. I mean, even measurement issues change over time. And so that is really interesting when you look at how people, for example, identify, because definitions even of race, ethnicity evolve for people over time. And particularly their definitions of sexual identity and their own gender identity. And that's why we are no longer exclusively MSM. We do have quite a few people who are trans now, and there are people who have transitioned. Initially, when we started mSTUDY, we wanted to include trans. We met with the community, and at the time, the community of trans folks in Los Angeles didn't want to be part of a study that was focused on MSM. Mostly, they wanted their own cohort, and we didn't have the capacity to do that. As things have evolved, as I said, some of our cohort members have transitioned, and also we now enroll people who are trans, and the community's understanding of that or interest in our study has changed over time. But like from the beginning, we were not enrolling trans by the choice of the trans community. So beyond the behavioral survey, we also do quite a bit of testing and could collect a lot of labs. So this is just a list of the labs that we test. We do this whole blood panel, which are CLIN labs. So we have all that done at every 6-month visit. We do testing for substance use. And I can go into a little more detail on that, but basically we do the urine tox screen at every visit, as well as we do the self-report. And we've used different tools over time, but now we are using the assist. And I'll talk to you about some upcoming things in substance use testing that is going on in collaboration with UCSF. We have done STI testing at every visit, and we test people. We do urine-based testing, rectal swabs, and as well as pharyngeal swabs. And we test for chlamydia, gonorrhea. We also test for syphilis. We did some HPV testing in the past, and we have stored samples on saliva and swab for that for the future. We do some clinical data collection. We do less now than we used to, but at the intro visit, everybody did get a physical exam. And now what we do at the 6-month follow-ups is with a clinician, we do a review of systems and we do medical history. So we have a lot of this data. We haven't used that much of it. If anyone here is interested, you're welcome. We do accept concepts. We love to see our data used. So we do have this very rich data that has been kind of underused. And then I mentioned the biorepository. So we established the biorepository from the beginning. We have now over 77,000 specimens. We are using them. We have quite a few studies and R01s and K awards that have been written to explicitly use our stored specimens. So that's very exciting. We have this range, as you can see, including the PBMCs, although we don't collect PBMC on everyone anymore, just on a select group. Because of the challenges of doing it, as you all know. And same with the saliva. We sunsetted the saliva and the nails, but we have quite a few of these still in the repository. So before I move to the cohort characteristics, I thought I'd just open it up and see if anyone has any questions about how the cohort is structured and what we do. Okay, so let me tell you what the cohort looks like now. So these are our participants. So this is across all visits from 2014 through, we closed this in 2023, this particular poll. And you can see age at last visit. So where we used to have quite a few that were under 30, guess what happens in 10 years? They age. So they are sort of now our mean is 37 for our people with HIV, and it's younger, 33 for those without HIV. You can see the median, and then race ethnicity. So we are, the cohort is about 40% Black and about 40% Latinx. We do have about 15% that are white among our people living with HIV, 9% without HIV. And then we have a few other ethnicities. What this doesn't really capture and show is that a lot of people are multiracial. And so we ask them to, what is the primary race ethnicity that you identify with? But we also have quite a few other questions on race ethnicity. So we do have a very diverse cohort. These are a little bit more about our participant characteristics. So you can see we have quite a few that are unstably housed. And so this has gone up over the years, actually. And so it's even higher when you look at last visit. But right now, it's about a quarter of those with HIV and 30% without HIV that reported unstable housing at the last visit. Depression, we use the CESD. About 50% of those with HIV and 40% of those without HIV. So we have a very sort of street engaged cohort. If you look at our employment levels, we only have about a third that are working full time across the groups, 15% part time, and then we have quite a few that are unemployed or on disability. That makes up 40% of those living with HIV and almost and over 30% of those without HIV. So if you look at our viral load characteristics, and this has really changed over time, and I was looking back through the data to do this. When we started, 50% of the cohort, it was about 47%, I think actually, were detectable. So they had viral load. We had quite a few that were viremic. In fact, we started by targeting and trying to enroll specifically people with virus back in 2014, and then decided that was going to be a challenge and that also people were going to move in and out of viremia, which they have. But at the last visit, and so that's why here is the last 2 years on the right, you can see we're down to about 26%. But when you look across all of our data, we are at about 35% of those that are considered detectable, having detectable virus. And there you can see what our median viral load was at last visit. And you can see again that this has gone down in the last 2 years over time. Our PrEP use has stayed pretty stable. So this is ever used PrEP. This doesn't really capture those currently on PrEP, but we have about 40%, and that's stayed really stable who've ever used PrEP. We've done a lot of work on PrEP, and I have to apologize because I'm actually not going to talk about that very much today. But we have quite a few publications on PrEP if you're interested that are out there. We've done a lot of work on STIs, and so I wanted to sort of show you where we are overall with STIs. So this is across, again, all visits. Really what's interesting is that we have a lot of gonorrhea, which is not something you often see. We have, if you look at any site, it's almost 9% across both groups. And we're usually, hovering around 5% at any visit. And we have quite a bit of early syphilis as well. So this is a population that continues to be at risk, even if they're on PrEP or if they have control viremia, living with HIV. They still have a lot of risky sexual engagement that you can see by, or high exposure by the amount of STIs we have. Substance use. So that's what everybody's interested in here. So that's what I'm going to talk mostly about. And so substance use by UTOX and self-report is actually compared here. So the light blue bars on the far left are self-reports, and the green bars are UTOX. So those of you, you're all here because you're interested in substance use. So you probably all realize the issue with comparing these two things and these two measures is the window, right? So we ask about substance use in the last 6 months at most of our visits, and the assist is the last 3 months. The self-report only captures for meth the last couple days. For cannabis, it can go back to a month, but this UTOX then has just a smaller window. So if somebody, for example, used on the weekend and they come to our study visit on a Thursday, they will UTOX negative. So we always get sort of a lower report by UTOX than we do by the self-report. But it does show you what is on board. And so when we do a lot of the basic science work that we've supported and the clinical research, we actually can see at that visit when we take those other assessments, such as the blood and the other specimens, we know what drug was on board at that time of visit. So here I wanted to point out we have a lot of meth use, both by those living with HIV that are the columns on the left and those without HIV on the right. And we've tended to always have more meth, around 30% above what we have for those without HIV. We also have, of course, we're a California state. We have a lot of cannabis use. And so that is our use here on the left. So this is over time where we've been with meth and cannabis use. And so if you can look, the blue bar is the meth use, and it's actually stayed pretty stable. It's come up a little bit. So what that is, it's pretty interesting. The bars on the far left are the 2 years, 2016, 2017, and actually 2018 were UTOX. So the lower numbers are probably because that was done by UTOX, not by self-report. And I didn't have time to pull out the self-reports from those years. All of our overall reports at our meetings were done by UTOX at that point. So I think some of that actually probably is not a trend. I think the trend is pretty stable that people have used, and we have done some latent class analysis for individuals, seeing that people have tend to be pretty stable in their use groups. And so I think that is pretty interesting. And you can see the cannabis use is the green bar, which is the highest and has stayed pretty stable. But right below that is, that's cannabis use for the people with HIV. And right below that is the meth use for people with HIV. That has also stayed very stable. So again, you know, I mentioned this thing about UTOX versus self-report, and I wanted you to see that actually we compared this. And so here you can see this over time, and you know, there's not that big of a difference, but you do always get a higher report by self-report than you do by the UTOX because of the difference in window. So I'm going to show you some findings now, but before I move on, any questions or comments? Okay, I'm going to keep going. So, oh, and by the way, I should say all of these images that I'm sharing here are images that are part of our study. So they're all part of our outreach materials, our recruitment materials, our study focus. And those have changed too, and I'll show you a little bit how those have changed. So the bottom line, and what I'm going to tell you is, what have we learned in 10 years? We learned kind of where we started, which was that meth, it's all really about meth. And whenever we look at substance use, meth just overrides everything. Meth, we've looked at, though, individually. We've tried to pull out just meth use and have found that it affects mental health, particularly depression. It affects sexual health, STIs, transactional sex, interpersonal violence. It affects other comorbidities. It affects HIV prevention and care. Whether or not people are on PrEP or how they do PrEP, whether or not they stay in care, whether or not they're viremic. We've looked particularly a lot at some of our basic science research has looked at the role of meth on inflammation for people with and without HIV. We've done a lot of work on the microbiome. And in fact, we're just awarded a oral microbiome piece, R01 as well. And so we continue the work on microbiome, but we have shown a lot of the interaction between meth and the microbiome in our work. And we've done some work on social genomics as well. And so those are all things that meth affects, but we have also looked at what affects meth a bit. And in looking at what seems to be associated with use of meth, and particularly possibly driving meth use, is things like homophobia and homophobic victimization in particular. And then also, some intersectional issues around racism, stigmatization, and social capital. So let me show you now some of these individual findings. So one of the papers that we did, led by my MPI, Steve Shoptaw, was really that he's always believed in these different phenotypes, if you've ever heard him talk, of meth use, that not everybody used meth the same way, and that, you know, there are weekenders and that there are daily users, and that that there's a lot of variation in how often people use, as well as sort of what much they use. So Steve and I have always been a great team, and one of the differences in how we look at meth is he's really interested in sort of these phenotypical patterns of weekly, and I'm interested in sort of the more contextual patterns, and also the actual, as an epidemiologist, exposure, how much there is. And so you'll see that sort of driving our work as we move along. But some of the first things that Steve did was to really look at the effect of meth use by the frequency of use across different comorbid conditions. And so as you see here, the dark bars are those who use meth the most. And the more you use meth, the higher prevalence we found of these other particular conditions. And this went beyond just clinical conditions. This also went to some of the sort of broader social conditions of how people lived. And so here you can see on the right, these are predicted probabilities of these adverse health outcomes, and sort of the same dose response relationship to meth use. And so here you can see like unemployment and housing instability. And again, the more people use, and those are those little diamonds, the more you see of these conditions. IPV is in here as well, new partners, exchange sex, STIs, all of these are significantly associated with this, as well as and have very strong relationships, as well as detectable viral load, STIs, renal conditions, neurological conditions, and psychological conditions. So I mentioned mental health and depression. We also looked at depression over time and looked at this again with the amount of use in more detail. And so here, if you look on the left, we looked across all the drugs, and we actually saw there was a lot of association with frequent heroin use as well as meth use. But those who used heroin and meth had the highest prevalence of these depressive symptoms as measured through the CESD. And then if you look on the right, you can see the predicted CESD scores across study visits. So this is over time by substance use. And again, meth, which is the top one, comes out on top. Much higher scores for depression among people who used depression. And declines in depressive symptoms over time was noted for most drugs, except for people who use meth, who showed no change over time. So as I mentioned, we did a lot of work on inflammation. And, I am not a basic scientist, and so I leave this work to my ID colleagues to really talk about. But we have seen methamphetamine use associated with rectal mucosa inflammatory cytokines and with STIs. That's most of our inflammation measurement is done through cytokines, and we have quite a few papers that have looked at this. And it's a pretty strong finding that has pervaded. I'm going to talk about this cannabinoid one in a little bit more detail as we move on. I talked a little bit about injection and how we didn't initially enroll people with injection, but after a couple of years of the cohort, I decided that we needed to take a look at injection because we were hearing that people were transitioning to injection in meth use. And I want to say this paper was published in JID a couple of years ago, and I am in the process of updating this analysis, and I'll tell you a little bit about that in a minute. What we found here, though, was pretty compelling. If you look on the left here, we found that people who inject meth, and those are the purple bars, more of them were living with HIV, more of them had viral load less of them were undetectable. So that means more of them were viremic, and more of them had any STI. So that's the left side. So there you can see that strong association with injecting meth, not just using meth. And the blue bar is using meth. So it follows behind the meth injection pretty closely, and but still meth injection really jumps out. If you look then on the right, you can look at some of the sexual behaviors that people reported who also were injecting meth. So the dark, again, blue, the dark here, orange bar is meth injection, followed by non-injection, which is the orange bar. And here, new sex partners, having transgender partners, having concurrent sexual partners, and then most notably on the far right, there's a big difference. If we look at people who receive drugs, money, or shelter for sex, so that in other words, engaged in transactional sex, much higher, almost twice the percent of people who are injecting meth are engaged in this type of behavior than in other people who use meth, and way higher than any other drug use. So we found that people who inject meth have viral load and STIs that can facilitate transmission of HIV. And combined with their high engagement in behaviors that include multiple ways that HIV can be transmitted or acquired, like transactional sex, these transitions to meth injection deserve attention. And that's why I decided to pay attention to it again and sort of see what was going on, especially as we've heard about a lot of fentanyl going on in the community. And, anecdotally, a lot of people talk about, oh, well, now people inject. So we pulled the data, and I was hoping also to present this today, but let me just tell you, it is way more complicated than we expected. We had heard sort of empirically that once people transition to injection, they continue to inject, and that's sort of where they are. what we found was very different, actually. If you look, people are moving back and forth. They inject for a while, then they don't inject, then they return to injection. And so we are going to have to use some more interesting methods of analysis to sort of show these internal transitions that people are going through. The pattern is not as simplistic as we thought it might be. And I think that that is actually really going to lead to a very rich finding, because we're going to learn what drives these transitions. And I'm going to show you some work and some methods we've done on other type of transitions, but I think these transitions are some of the benefit of doing cohorts, is that you can see the factors that move people between these different states. I mentioned IPV. This is a recent paper that we had that was done by one of our T32 trainees. Here we looked at, started by overall substance use. And here I love to see these nice models. You can see that this here shows how this was seen to be associated with an IPV. And what we found was that it wasn't just drug use. It was specifically, of course, again, stimulant use, which here is our meth use, which was associated with IPV. THC was not, and other drug use was not. And this is, of course, adjusted for age, race, ethnicity, transactional sex, depression, serostatus neutral HIV prevention, partner type, and homelessness. And this is all IPV in this particular figure, but if you go to the paper, you'll see we actually looked at the different types of IPV: sexual violence, physical violence, and mental violence. And we saw these relationships across all types of IPV. So I mentioned microbiome. We have done quite a few papers on microbiome. But I wanted to just sort of pull one out and tell you that this was actually done by one of my doctoral students, who is now on faculty up in Oregon, that we found that HIV was associated with dysbiosis in the gastrointestinal microbiome in a dose-dependent manner, and really pulled out, and I have some of those beautiful, heat maps and complicated drawings of microbiome, but I'm gonna not go into those today. Let me see if there's something in the chat. Hi, Elise. Yes, and it was nice to see you. Did I adjust for frequency of meth use when we looked at meth injection? No, I did not yet, but we can look at that in the upcoming analysis. So it was injection as a mode of use, not as a frequency not as a surrogate for frequency. So frequency was assessed separately, so we could look at frequency along with the mode of use. I hope that answers that.
Yes, thank you.
So then I'm going to sort of wrap up the work we did on meth, although we have done tons. So this is just sort of snippets by this one really interesting paper that Jen Fulcher, who is a colleague on this, she had a K award on the mSTUDY, and now she has 2 R01s on it, one on the oral microbiome, and what's the other one on? I can't remember. She just got a second one. Inflammasomes or something. But Jen, you should have her come talk to you as well someday. She's a clinical researcher and a virologist, and she has really done a lot of work on this. And she did this really interesting paper looking at the factors associated with viremia among their participants. And these are participants on ARVs taking therapy. And what she found was that, of course, it was methamphetamine only that was really strongly associated with having viremia, but also meth use with other drugs. And when we excluded meth use and looked at other substances, there was not the same association. So meth use, either alone or with other substances, was associated with the higher odds of this. And that we could see that among the meth users, nearly half of the instances of viremia would be reduced if meth use was discontinued. And that was the attributable fraction of 46%. So meth, it's really all about meth here. And I can talk more about meth, but I'm going to now move on and talk about, if my computer will let me, Oh, no, I have some final ones. I forgot. So I did want to say that we do look at things sort of more intersectionally and try to understand sort of the context in which meth is used and what's happening here. And this is a paper done by somebody who was, again, a T32 trainee at the time and is now on faculty, where he looked at homophobic victimization, housing, and meth use, and saw that, you know, this homophobic victimization was associated with a greater severity of meth use. And this was just across a 2-year period that he sliced. And that it was also associated with a greater duration of being unstably housed. But that unstable housing itself partially explained the association between this homophobic victimization and meth use. So that these, what I like about this particular paper is that it shows that these relationships are more complex and that there is a lot more involved in what actually drives meth use than we know, and that has to do with the context of how people are living and their personal life experiences. The other paper we had that sort of looked at this more broadly, again, was looking at social capital and also experiences of homophobia. And here, one of our colleagues looked at how having capital resources and experiencing these affected whether or not people were engaged in HIV transmission behaviors, and also in testing, engagement in care, and ART adherence, and virological suppression. And if you see on the right, you can see that again, although we have alcohol use in here, really we have a big role of meth use during sex. And that continues, and this was meth use during sex, not just meth use across this. Finally, again, Michael Lee, who was one of our trainees and is now faculty, was interested in social genomics and wanted to look at this one thing some of you may know of, which is this type 1 IFN, which again, don't ask me to talk about it, but it's a gene expression. And he looked only among those that were Black and Latinx MSN in here. And what he found here was that methamphetamine use and unsuppressed viral load and experienced IPV additively predicted inflammatory and type 1 FN gene expression among minority MSM in our cohort. And this demonstrated that social adversity can upregulate an inflammatory gene expression here, and also demonstrated independent contributions by methamphetamine viral load and experienced HPV in launching these inflammatory cytokines. So this was sort of a unique piece of more genomics work that was done in this. But now I'm going to leave meth, and I will stop and see if anyone has any questions. That was kind of a grand tour of meth, and I'm going to talk a little bit about cannabis. Any questions? Okay, let's talk about cannabis. So remember, we had a lot of meth and we have a lot of cannabis. And I have to say, you can't study meth without studying cannabis, because all of our participants who use meth also use cannabis. But you can study cannabis without meth. And so with cannabis, we have looked at cannabis on mental health, cannabis effects on sexual health, a little bit on social genomics, cannabis on HIV progression and inflammation, cannabis and microbiome, and cannabis and HIV prevention and care. And one thing I want to say on inflammation is we're very committed to this work on cannabis and inflammation among people living with HIV and without HIV, because there's so much stuff out there about cannabis and inflammation that it is anti-inflammatory, and whether or not it is and in what situations it is something that we want to understand a little bit better. But when we look at inflammation, I want to be clear that we look at it at the cellular level. We're looking at inflammation through the cytokines, and we're not really looking at inflammation clinically. So we don't know if people are feeling differently, and that's another whole area of research that I do think is important, but not something we've taken on yet. And again, we do look at homophobic victimization and other issues here. So this is a paper that I did a couple years ago now that, again, we're hoping to update, but I was interested in associations between cannabis use and sexual behavior and STIs. This study here, I looked at the percent of participants here who were reporting substance use and cannabis use across visits when we had about 3 years of data here. And so here you can see I divided people up into these buckets, and we have people who didn't use any substance use, although we had some binge drinkers in there, but not very many. And then we had people who only use cannabis, people who used no cannabis but other substances, and then people that used cannabis and other substances. And then when we looked at this, what we saw was really kind of a dose-response relationship in the opposite way that you would expect when we looked at the prevalence of STIs. And so here, these were our STIs, which were gonorrhea, chlamydia, and syphilis across all sites. And we found that those who had STI at a particular visit, and we looked at that with the use reported of cannabis at that visit, the more they used cannabis, the lower prevalence of STIs they had. So you can see on the left, the really strong relationship here. Those who are using weekly or more cannabis have much lower STI, only about 4.7%, compared to those using monthly or less often. That's 16%. And it works the same way with, if you look at the other substance users, it goes the opposite direction. So the more you use other substances, which include our meth, the more STIs you have. But cannabis seemed to be working the other direction. And so a funny story with this paper is I wanted to call it Safe Stoners. And the journal wouldn't let me call that because, what we really, this was around the time that cannabis became legalized. What we really felt this shows is that, these folks who are heavy cannabis users, they're not really engaged in street drug culture in the same way. We also found in the same paper that they had fewer partners that were incarcerated and had less IPV and had less sex in general and less risky sex than other folks. So the more they smoked cannabis, the less they were doing sort of these broader risky drug-related behaviors. So that's the conclusion was fewer MSM reporting only cannabis use than other drug use, had been incarcerated, had incarcerated partners, experienced IPV, and were living with HIV. In our multivariable analysis, we found positive STIs were associated with other drug use, but not use of cannabis only, or no drug use after controlling for age, HIV status, new partners, and number of sex partners. So when MSM reported using cannabis exclusively, fewer STIs were detected and lower risk sexual engagements were reported than when MSM reported no drug or other drug use.
Can I ask a question?
Sure.
Thanks. This is just so interesting. Were you able to look at this causally? Sorry, I didn't quite catch if it,
Yet.
Okay. So it could be just their personalities or their cluster of behaviors.
Yeah, but that cluster of behaviors we felt like was important. So that's why we tried to look at it. It was at visit. So it was at each visit. It wasn't like a lagged.
Yeah.
So we could do it lagged. Or I
just
can
curious.
give you and you can do it.
No, sorry.
But we're going to do more. This was early.
It's fascinating. So I just wanted to see where you're at with it. Thanks.
Yeah, we want to do more with cannabis, and we will when we stop writing proposals and start doing more analysis. Everyone knows that game. Okay. And so this is also something, as I said, we did some basic science work with cannabis, and this is out of my lane, but I wanted to show this for people here who are. There are, this was a study done by a postdoc we work very closely with our Cannabis Research Institute, Ziva Cooper, who heads it, has been a co-investigator on a number of our papers and even on a small grant. And here we looked at changes in immune-related biomarkers and endocannabinoids as a function as changes in cannabis use frequency in people living with and without HIV. And on the right, you can see these two immune markers here. And without HIV are the black boxes, and with HIV are the white ones. And so for this TNFR2, it was lower for people with HIV, and there was no difference in frequency of use. And when we looked at this CD27, you saw the same thing, lower in people with HIV than with people without HIV. And then he looked at these other things, which are AEA, which those of you interested in this probably knows what those are, that are regulated by this FAH, F-A-A-H. And here we saw the opposite relationship with those with HIV had more of this than those without HIV. And so the bottom line was that people living with HIV exhibited greater levels of the other ones that I showed you and lower levels of these, and that frequency of use did not impact the immune phenotype or endocannabinoids. So this was on sort of a small sample, but this was sort of a dig into what's going on in terms of with endocannabinoids. So we have a lot of other work on cannabis, but a lot of it is sort of more basic that goes way beyond my lane. So I'm not going to talk about that. But what I do want to talk about is this really exciting new R01 that we're doing with you folks at UCSF. And I see some of the members of this team on this call, and that's great. And so we need a cool name for it. I call it the hair study. But this just shows you how long and productive relationships can be in doing this work. And back in 2014, I originally reached out to your colleague, Dr. Gandhi, and asked her about hair and said, I want to look at hair for substance use and I want to measure it. And at that time, she did not have an assay to do that. So I said, you know what, I'm just going to collect it and store it. And so since 2014, we have been collecting and storing hair. And finally, last year, she was able to put together the R01. And so with mSTUDY, we are doing this really exciting R01 to develop this assay to look at ways to measure and actually quantify substance use in hair samples. And so I'm sure you've all heard from the work that Hal does, but basically this will be able to show us through segmented analysis the real quantity and the amount of substances that people consumed back over to, we hope, about 6 months, by month by month. And here we'll be able to examine the association with this once this method is developed with self-reported use and urine screens and clinical outcomes, you know, so that we can combine these different measures and come up with ways to really narrow down the level of exposure and also help us interpret these self-reports and the UTOXs so that we can use them as proxies for other things. So this made some great progress. And another one of your colleagues, Aisha Abba, is doing a poster at Croy on the preliminary findings from the hair analysis, and it does show that we so far detect more meth use than we did either with our UTOX or self-report. So that's really exciting. And finally, we're going to be able to use this more rigorous assessment of the levels of substance use in looking more tightly at this issue of systemic inflammation. And as you know, that over the course of mSTUDY since the beginning, we've been interested in studying inflammation, whether it's rectal inflammation or systemic inflammation, and now looking in other ways, we're going to finally be able to have a good measure of exposure by being able to use the hair reports. So by the end of the study, we hope to have developed and validated metrics for substance use in hair to provide robust and objective biomarkers of substance use for NIH studies, examining interventions to reduce substance use among people at risk of or living with HIV, and the association between substance use and inflammation. So super excited about this study. But we didn't stop there. And we actually said, okay, oops, I did click the wrong thing, hang on. We actually said it's time to do more with this. And so we put in a proposal, just again with the support of Hal, and leveraging this really exciting new R01 to look at what we're calling this new study ELAPS, which stands for Effectiveness of Long-Acting Injectables for People Who Use Substances. And these are long-acting injectables just for ARV, for HIV treatment. And we want to look at this using a biobehavioral approach in mSTUDY to look at how people with HIV who use substances, how viral load and inflammation differ by those who are using long-acting injectables. So since this is a proposal and we don't know if we're going to get funded, I'm not going to say too much more about that, but we're really excited that this is the kind of work we'll be able to do now that we will have this hair assay. And that takes me to, I'm pretty much on time here, we're evolving. So after 10 years of being an observational cohort, mSTUDY has gotten to the point where we want to do interventions and we want to see a little bit more about what interventions can do for people who use drugs. And so as part of that, I wanted to show you this paper that was done by my doctoral student, Ali Rosen, where she looked at the effect of treatment on methamphetamine use. And as all of you know, there is no medication really for use for methamphetamine treatment. And so this was using treatment that people reported across a whole range of different types of treatments, from inpatient treatment and outpatient treatment and being in support groups and any kind of treatment And there was a big range. And what she did was really focus on these transitions, these what she called sojourns, which were the time people spent in these different states. So using a really unique type of analysis that I'll tell you a little bit about, her goal was to understand how people moved across these different states in the mSTUDY. And here's what she found, which was really, I think, amazing, given this is a community cohort that we're observing this, is that people moved across different states who'd had treatment in ways that we found sort of powerful and surprising. So I got to move my thing so I can describe this to you. So meth use, people who moved to no use is the bar in the far left, and then to occasional use is this darker bar, and finally to frequent use is the light bar. And so those who received treatment are this box on the left. And what you can see is that people who received treatment more moved to these lower use groups than to the frequent use group. So being in treatment made a difference. And if you look at those on the right, these are those who did not receive treatment. And you can see that fewer of them were able to transition to occasional use or to no use than they did if they were not receiving treatment. So this is a pretty, I think, profound fire. And if you look from frequent use, actually, you can see even the biggest difference here. As those who didn't have any treatment, that we saw more people transition to frequent use and much fewer transition to frequent use among those who received treatment. So this finding, showed us that this was done with dynamic modeling, that shows us a new method for assessing these longitudinal methamphetamine patterns with these multi-state models. And added important evidence outside of clinical trials that substance use treatment may reduce methamphetamine use. We're also doing this other really interesting intervention on mouthwash, which has been shown in some studies to prevent pharyngeal gonorrhea among a high-risk population. And so we're trying to see if that does make a difference. And so we have a randomized control trial where we're giving mouthwash out. It's a really hard study to do, but people are using mouthwash that has antibacterial properties as well as those that don't. That's the Listerine versus the placebo. And we're going to see where this goes. And then I'm getting close to filing up, and I wanted to just show you this model that I actually have just developed, and that sort of, I think, encapsulates sort of the ways the mSTUDY wants to look at things in terms of how their substance use intersects with HIV and how biology and behavior both work together. And so what we do is we look at sort of, yes, we recognize the importance of individual characteristics, and beyond just people's race ethnicities, it's their experiences of racism. And housing stability and income stability, and also their partner characteristics. And I didn't show any of those papers here, but that's something we've actually looked at a lot, and that's always been a personal interest of mine, is the role of partnerships. How this and mental health then interact with substance use. And when we look at substance use, we want to look at substance use in a much more nuanced way than just frequency of use or the type of drug people use. And while those are both in here, we also are doing more to look at these patterns of use, comparing episodic use with continuous use, looking at the mode of use, whether it's injection or not, and then the context of use, whether people are using drugs in social situations, during sex, or for trade or survival situations. And that these things together really affect the drug levels that we see in people. And that, of course, substance use treatment, we do believe, can impact this, as well as adherence to ARVs, both long-acting and not, and also PrEP. That these affect the drug levels for people living with HIV, and also ultimately inflammation and viral load. So this is sort of an overview of a model of how we feel like substance use and HIV intersect in the lives of these men that we follow. And then I'm going to finish by showing you some of our fun different images we've used over time in mSTUDY. I want to say the far left we did a photo shoot at the center years ago, and these are real community members who, you know, we live in Los Angeles, everybody wants to be a model, and so a lot of them are. And so we did this photo shoot where people came in and we took these pictures for it because we wanted our images to be from the community, whereas the ones, the other ones you see are more stock photos. But these were considered a little bit too sexually exploitative, some of our participants said, so we toned it down a little bit. And we have on the far right are some of our most recent examples of images that we have. And in the middle, you can see some of our study team from our last annual meeting. These are both across the sites and some of our researchers who were able to come out. And they really have been an incredible team. And finally, I really do want to give a heartfelt thanks to the mSTUDY participants. They are our inspiration and the raison d'etre for this work, and we strive to support them in improving their health and life experiences. And it is tragic to have seen so many that we lost. And everyone that is lost, we contribute to the Project Angel Food to honor their lives. So I will stop there, and I have a few minutes for questions.